Bismillahirrahmanirrahim. Our today's topic is about genetic engineering, which is included in your course. And actually, genetic engineering and biotechnology are interconnected. So that's why we will be discussing about genetic engineering and biotechnology together. But our main focus will be on the genetic engineering. So first of all, what is genetic engineering? Usually, when people ask me that what is genetic engineering and what is biotechnology, so usually I told them that a biotechnologist is actually a medical engineer. So it is the combination of both medical and engineering. So in genetic engineering, what we do is usually in engineering, what we do, we replace or add or remove some specific parts from some specific machine, or improve that to improve that machine, or improve its quality, or maybe to reduce its size or something else. So basically, it is engineering. In genetic engineering, what we do, we basically remove or add or suppress or improve the gene to gain a good quality of crops, or a good quality of animal. Like for example, cows. Usually, if you have heard about the hybrid cows, this is called this is basically the phenomena of genetic engineering. So in this lecture, we will be discussing three different topics. The first one will be recombinant DNA or genetic engineering, and the second will be applications of genetic engineering, which includes research on humans, as well as research on animals, research on plants, and research on microbial genes. The third portion of our lecture will be the release of genetically engineered organism, which will include biosafety and ecological applications, potential ecological concerns, and regulatory policies. So starting with genetic engineering, first of all, what is genetic engineering? Genetic engineering is the technology that involves manipulating the DNA of one organism in order to insert the DNA of another organism, called the exogenous DNA. So what we basically do in genetic engineering, we basically add or insert a DNA or remove the DNA from a specific organism, and then add that very much specific DNA to another organism in order to produce a good quality, or improve some specific quality of an organism. So this is actually called as genetic engineering. Here is the definition of the genetic engineering. Genetic engineering, engineered organisms are used to study the expression of particular gene. Like what we do, we usually genetically engineer an organism to study like what was the gene responsible for, and the second thing is to investigate cellular processes, or we can also study about that specific cellular process, like what was the role of that specific gene in any cellular process, or if we remove specific gene from an organism, what will be the result. Study the development of a certain disease. So it is also important in studying a specific disease. Like for example, if we add or remove a gene from an organism, like if you want to study that either is this gene is having some role in any type of disease or not. So this is also included in genetically engineered organisms. Select traits that might be beneficial to humans. So as I mentioned earlier, that we usually produce specific quality of crops and animals. So these are selected traits or specific traits or good quality traits which we usually produce which we usually produce for humans, so that they can provide us a good quality of crops. Usually in animals, what we do, we are in need of good quality of milk and good quality of meat, and other different types of traits as well. But usually we are in need of on very basic level and very low level, we usually prefer good quality of milk. Like we usually do genetic engineering to produce good quality of milk. So good quality or quantity as well, like not specifically the quality but quantity as well. So this was all about the use of genetically engineered organism. Now we will be discussing the biotechnology. We have already discussed, but we will be discussing biotechnology and genetic engineering together to understand the difference and similarities in them. So the use of living cell to make products such as pharmaceutical food and beverages. So like in biotechnology, what we do, we make good products with the help of genetic engineering. Like for example, if we are in need of some specific drug or some specific medicine, so we use genetic engineering technology. So basically genetic engineering is a type of technology, and bioinformatics is the study or performing that like genetic engineering is basically a type of tool, you can say. And biotechnology and biotechnologists use that tool for different types of activities, like for example production of good quality of foods and drugs and beverages. The use of organisms such as bacteria to protect the environment. So what we do, like they have mentioned over here the use of bacteria, but we also use different types of other microorganisms for the study of different types of you can say for the production or the use of different microorganisms for humans. The use of DNA science for the production of material for diagnostics and research. So what we do, we use DNA science, like studying the DNA and the qualities and characteristics of DNA for the production of material for diagnostics and research to study for example to study the basic basic production and the results and the diagnostic tests which we usually perform with the help of DNA, like for example ELISA, PCR and the other strip technologies as well. And also we use that DNA science for a research purpose as well. Biotechnology helps to meet our basic need, like for example food, clothing, shelter, health and safety, etc. So biotechnology usually focuses on the basic need. Like they also have another properties and qualities as well. Like they also have the purpose of research other than these as well. But the first and basic development and the need of development and the need of research is with the very basic needs, like for example food, shelter, clothing and safety, etc. You can see in the image now, I know how to enhance the quality of my life. So after studying the biotechnology, we are able to know that how can we enhance the quality of life. Now what is recombinant DNA? The manipulation and combination of two DNA from two sources. The manipulation and combination of DNA from two sources. What we do in recombinant DNA, we usually add two different DNAs from two different sources. For example, if we extract the DNA from plant and the other DNA from another plant, or either we can extract the DNA from bacteria and add that very much specific part of the DNA to humans. Like for example, in the use of insulin, what we do, we extract the insulin gene from humans and then add it to the vector of the bacteria, and then when the bacteria divides, the gene also divides and produce insulin in much for example much more requirement, much more you can say quantity. So bacteria DNA plus human gene for insulin. So when we add these both, so this is called recombinant DNA. And plant DNA plus bacteria, agrobacterium tumefaciens is a good example as well. mouse DNA plus human is equal to transgenic production. Usually if you remember, we have discussed about the transgenders. What actually occurs over here is the same, like mouse DNA and human DNA. So these are different and DNA is from different like animals, so that's why we call it transgenic. Genetic engineering, so what we do, insert a foreign gene into a host. What we do in recombination, like it is actually the recombination. Re means again and combination means combining. So what we do, we remove that gene and then recombine together for what is our goal over there? To produce many copies, clones of a particular gene. So this is our goal, that we need multiple copies of a gene. So what we do in recombinant DNA technology, the first thing is that restriction enzymes recognition sequence. Like first we cut two specific genes with our desired restriction enzymes, and then in the second step, addition of DNA fragment from another source. If I mentioned that in both of the organisms, in the both of our desired organisms, like we extract the DNA from one organism and then add that specific part of the DNA to another. So what we do, we actually cut that specific portion of the DNA with restriction enzymes, like both we will cut both with the same restriction enzyme, and then we will add that to specific then we will add them together, and after adding DNA ligase, paste the strand together, and then recombinant DNA molecule is produced. Now in the next step, what is the recombinant DNA molecule is produced, and now we get that very much recombinant, that very much specific kind of molecule which we were in need of. So in the next step, what we do, so here are the vectors. Vector is an entity that is an entity that transmit a disease or parasite from one animal or plant to another. So what is the vector? It is actually an entity or that transmits. Vector is actually a part of a DNA, or you can say, and we can also use animals and viruses and different particles and DNAs as well, that transmit the disease or parasite from one animal or plant to another. So we have, if you if you remember that we have already studied vector in previous lectures, that vector is a part of a DNA usually present in bacteria, which is called bacterial vectors, or we have already studied. But vector can be present in different organisms as well, like we were discussing about bacterial plasmids over there. So inshallah we will continue this lecture after inshallah a while. After that, because the timing is not